Hello, everyone, and welcome to today's webinar. My name is Chris Mathis, and I'm Chief Commercial Officer of AnaBios. AnaBios is a unique contract research organization and biotech company based in San Diego. We procure human tissue samples from ethically consented donors and use this tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first-in-human. AnaBios has developed this webinar series with experts in the field who we admire and or collaborate with in our preclinical drug discovery research platform. These are topics that interest our scientific team, so we figure that they would also be of interest to our partners and network connections. So it's my pleasure to introduce today's distinguished speaker, Professor Cesare Terracciano. Hello. Professor Terracciano is Professor of Cardiac Electrophysiology, Director of the BSc course in Cardiovascular Sciences at Imperial College, and PI of the British Heart Foundation Cardiovascular Regenerative Center. He graduated in medicine at the Università La Sapienza of Roma, Italy, and qualified in cardiology in the same medical school. He obtained his PhD in physiology at the National Heart and Lung Institute in London. In 1998, he received a Wellcome Trust Career Development Fellowship, and his work is now funded by the Medical Research Council, the British Heart Foundation, the Wellcome Trust, and the NC3Rs. In his career, he was one of the first to study the mode of regulation of cardiac calcium transporters, the effects of manipulating calcium transporter expression using transgenic technology, myocardial and microdomain plasticity in response to mechanical unloading with left ventricular assist devices, characterize modalities of application and the paraffin effects of cell transplantation, and defining the electrophysiological properties of stem cell-derived cardiac myocytes, their chamber specificity, and their use in tissue engineering. Professor Terracciano's main interest is the electrophysiology of heart failure and the consequences of treatment on the contractile and electrical properties of the heart. His research focuses in particular on the mechanisms of myocardial regeneration following mechanical devices or stem and gene therapy, and the cellular and molecular mechanism arrhythmias. This particular webinar is very interesting to AnaBios scientists because we have great interest in developing a cardiomyocyte platform with our human cells that can be used for chronic applications. And so, again, it's my pleasure to introduce the presentation today entitled Myocardial Slices as Novel Discovery Model to Study Biology of the Human Heart.
So I was invited to talk about myocardial slices, a novel discovery model to study biology of the human heart, but also these applications to look for novel therapeutic targets. In order to deliver this webinar, I've asked my collaborators to help me in the delivery. And these are Fortius Vitulis. Fortius, would you like to introduce yourself?
Hello, my name is Photios. I am a medical student doing my PhD in Cesare's lab.
And then I have Barrett Downing.
Hi there, my name is Barrett Downing and I act as Cesare's lab manager and research technician.
Thank you, guys. That help is very important to ensure the smooth delivery of this webinar. Let's start.
One would be inclined to think that in 2019, after centuries of well-funded cardiovascular research, we would know everything about the biology of the heart in normal conditions and during disease. However, while our knowledge has increased substantially, cardiovascular disease remains a pressing problem, as it is one of the major killers, affects quality of life, and is a significant economic burden for health services. Cardiovascular disease discovery and its treatment is a story of two tales. while in the last 50 years acute mortality for cardiovascular events has been reduced substantially, surviving patients develop heart failure with huge numbers of hospitalization and very poor quality of life. There are several reasons for the discrepancy between research investments and relatively poor outcomes. The massive complexity of ultrastructure and regulation of the cardiovascular system are certainly essential factors. Equally, the lack of effective experimental models make the testing of hypotheses and accumulation of essential knowledge very difficult, in certain areas impossible. In this talk, we will describe the ideal experimental models to study cardiovascular biology, the most common existing experimental models and their pitfalls, and how they compare with the ideal model. We will then present a novel model that we have adopted in our research and that is gaining popularity, the living myocardial slice. Cardiovascular research is either performed using clinical trials, which lack mechanistic insight and are difficult to refine, or using reductionist studies with animals or cell systems, which are difficult to translate into clinical practice. It is extremely difficult to bridge the gap and study cardiac tissue from patients with sophisticated in-depth laboratory techniques to determine pathophysiology and novel pathways for treatment. This is not only due to low availability of human cardiac tissue, but also to lack of techniques that allow physiological measurements from intact multicellular preparations in a chronic long-term fashion. For this reason, the ideal situation where cardiologists, surgeons, physiologists, and basic scientists work side by side to resolve a common question is rare and confined to very few outstanding research centres around the world. The perfect cardiac model should mimic physiology and disease, as it occurs naturally in vivo, faithfully resembles human myocardial biology, and allows high throughput experiments and mechanistic studies. Desirable properties are that it is of human origin, that it has the ability to maintain in vivo features in long-term culture, it maintains the features of complexity and maturity, it has the ability to sustain normal and pathological workloads, it has the ability to be studied at multiple levels, for example, tissue, cellular, extracellular, and the molecular level, and it should be low cost and easy to use. The perfect cardiac model does not exist, and our knowledge is derived by the use of several established models. None of these are sufficient, resulting in the need of several complementary studies. Human cardiac tissue of sufficient quality and size is only available from cardiac surgery. Endomyocardial biopsies taken using percutaneous procedures are often too small to be used for sophisticated laboratory techniques. For ventricular myocardial studies, tissue can be taken from explanted hearts during cardiac transplantation, or from apical cores removed during the implantation of left ventricular assist devices. For more than 20 years, thanks to our close collaboration with the Harefield Hospital, many groups at Imperial College have performed studies using isolated ventricular myocytes and shed light on the mechanobiology of heart failure. However, the harsh isolation method, the lack of multicellularity, and the extracellular matrix, and the inability to maintain myocytes in long-term culture has limited our ability to optimize the use and significance of our studies. A human model of intermediate complexity suitable for long-term studies is needed. But what about living animal studies? In the past, these were the essential platform to study cardiac physiology and disease, and with the advent of transgenic technologies in mice in the last 30 years, have enjoyed a second peak of popularity compared with in vitro models. However, with the current stronger ethical concerns and the realization that there are very important differences between laboratory animals and humans, there is a strong trend to limit these studies only to the latest stages of the discovery process, and there is a concerted effort to find alternatives. The most common in vitro cardiac models are the retrogradely perfused whole heart, called Langendorf, isolated papillary muscle and trabeculae, and enzymatically isolated cardiac myocytes. These models cover different biological aspects. Cardiac myocytes are very commonly used. They are relatively inexpensive, can provide high throughput, can be used to test mechanistic hypotheses and can be prepared from human cardiac biopsies. However, they lack the fundamental interactions with other cells and the extracellular matrix. They are often used in unloaded conditions and therefore don't mimic physiology faithfully and dedifferentiate in culture. There are also concerns that the enzymatic isolation procedure may modify their structure and function. In recent years, with the optimization of protocols for efficient differentiation of human pluripotent stem cells into cardiomyocytes and their incorporation into scaffolds to form engineered cardiac tissue, we have seen a revolution in modeling for cardiovascular biology. These models are of human origin and, by using iPSCs, can be patient-specific, can provide high throughput, and can be maintained in culture for several months, opening new avenues for much-needed chronic studies. However, we now know that pluripotent stem cell-derived cardiac myocytes in vitro lack fundamental features of the adult myocardium, substantially limiting their translational potential. Myocardial slices may represent a suitable model of intermediate complexity for cardiovascular research. Myocardial slices are 300 micrometer thick sections of myocardium with preserved structural and functional properties. Myocardial slices have been prepared from several species, but most importantly, from human samples. This figure shows a slice prepared from rat left ventricle, subsequently trimmed to obtain a homogeneous tissue with aligned fibers. Ventricular tissue is placed with the epicardium down on the stage of a vibrotome. The settings are adjusted to obtain a very slow advancement of the blade with the z-axis deflection of less than 1 micrometer. Approximately 30 to 40% of the cells in the external layers are damaged, but the internal layers are intact. We calculate that only 4 to 5% of the total number of cells in a slice are damaged. The electrophysiological and contractile function of these slices are remarkably good. The top panels show the slices mounted on a force transducer. The generated force reflects the species properties and the disease state. We measure calcium cycling using optical mapping and fluorescent indicators. The slices can be easily used to study conduction with multi-electrode arrays. These pictures show that the complex multicellular structure of the myocardium is preserved in slices. Panel A shows staining for caveolin 3 and connexin 43. Panel B shows the intact sarcomeric organization. Panel C shows the intricate capillary network. In D, electron microscopy can be used to study the ultrastructure, while in E and F, slices were studied for three-dimensional arrangement of vessels and the extracellular matrix. The 3D structure can be readily studied using methods that make the myocardium less refractive. This figure is from our scientific reports paper, where the tissue in slices was made transparent using the fast clear method. Both micro and macro vessels can be observed in 3D. A crucial problem for cardiovascular research is that adult myocytes, or cardiac tissue, quickly dedifferentiate in culture. Slices are not different, and when they are kept in culture even for a few hours, their function and structure revert to a less specialized phenotype. This intriguing behavior makes the slices an ideal platform to study myocardial plasticity. The remarkable change in phenotype occurring in culture is an indication of myocardial plasticity, is observed in every in vitro model, and is due to the change in environment. Simple static culture conditions cannot recapitulate the microenvironment of the beating heart. There are many factors involved in vivo, but the most striking and peculiar one is continuous and relentless electromechanical stimulation. We developed a stretcher system and applied preload over a physiological range of sarcomere lengths 1.8 to 2.4 micrometers, while maintaining essential factors such as electrical stimulation, oxygenation, and a continuous supply of medium. We found that structure and function of rat myocardial slices after 24 hours culture are optimally maintained at sarcomere length of 2.2 micrometers. This slide only shows data for contractility, calcium transients, and T-tubule structures, but we found similar results for conduction, connexin 43 expression, beta-adrenergic response, arrhythmogenesis, cardiomyocyte morphology, and stromal cell population. We performed RNA-seq on samples cultured at 1.8 micrometers unloaded and 2.2 micrometers normally loaded, and we found that unloading induces downregulation of genes associated with cardiac structure, function, transcription, and translation, and upregulation in genes associated with inflammation and ECM remodeling. Genes encoding for sarcomere structures, such as MLP and TCAP, are upregulated in the normally loaded group, suggesting the importance of these structures as mechanosensors or effectors in the maintenance of the adult normal myocardial phenotype. We could repeat these observations in samples taken from human biopsies. We could also maintain contractility in rabbit slices for up to 5 days in culture. Several other groups have been able to maintain myocardial slices in culture. Using an oxytonic condition, the group of Andreas Dendorfer in Munich could maintain human cardiac slices beating at 0.2 Hz for up to 4 months.
Another application of the use of myocardial slices in the context of mechanobiology is to use a dynamic electromechanical stimulation. This is a project developed by Fortis Fitulis that will take over to describe this application.
So in looking, in studying remodeling with slices, current approaches have either used an isometric mode of contraction, in which myocardial slices are loaded on stainless steel stretchers and then stretched to preload by rotating a screw on the side. This allows the tissue to contract in an isometric mode, which means it's not shortening. This is a technique that we developed in our lab, and the production of force depends on the sarcomere length. So for example, in those pictures, you can see a slice that is stretched to sarcomere length of 2 micrometers, corresponding to preload, and a slice stretched to 2.4 micrometers, corresponding to higher preload. So using this approach, we can simulate high or low preloads and allow the slices to beat isometrically. In other laboratories, they have used an oxytonic mode of contraction, in which the tissue is loaded on inflexible posts on one side and a flexible post on one side and an inflexible post on the opposite side. When the tissue is stimulated to contract, it shortens, and the shortening of the tissue and development of force depends on both the sarcomere length, that is the preload of the tissue, but also the stiffness of the flexible post. If we look at those two modes of contractions at the force length plane, they would look something like that. So an isometric mode of contraction on the top panel means that the tissue is producing force without any change in length. In contrast, an oxytonic mode of contraction means that the tissue is producing force while also shortening, and the relationship between force production and shortening is linear. Now, basic physiology dictates that none of these approaches are actually representative. In vivo, what's happening is a very fine sequence of events during the cardiac cycle that consists of changes in both force and length in a very intricate fashion. So during contraction, there is an initial mode of isometric contraction where the tissue produces force without changing length. This is followed by ejection of blood into the vasculature, where the tissue is shortening while also producing force. Subsequently, the tissue relaxes under constant length and then fills again with blood during diastole. Under this mode of contraction, it forms a loop, and we call this a force length loop, or in vivo, a pressure volume loop. Now, you would appreciate, if you compare this panel to those panels, that the current modes of simulating are not representative of in vivo physiology. So what we've done is we've developed, in collaboration with Professor Peter de Tom, a physiological mode of contraction for myocardial slices. This is based on a mathematical model which simulates the pressure on the heart and the flow out of the heart based on a resistor, based on two resistors and one capacitor. These are parameters that we can simulate using our model, and by doing that, we can simulate a range of conditions. When we do that in the system that we had developed, the tissue beats, as you can see on this side, but also shortens. And the pattern of shortening can be modulated by changing and adjusting those parameters. If we plot that in real time, on this, as you will see on this panel, we can have slices performing work loops as they would in physiology or pathology. So in this particular case, the first loop is a slice that we've exposed to physiological load. Then we can simulate also high conditions of high afterload by changing the parameters. We can also expose slices to high preload and therefore mimic conditions of volume overload, as you see over here. And by doing that, we have enabled slices to perform work loops under both physiological and pathological conditions. Now, to do that, we have developed a novel automated device which allows slices to be cultured in the long term while also performing the work loops. So we will go on to present you this device. It looks something along like this. These are the 3D renderings. The device consists of both hardware and physical elements, which you see on the top left over here, as well as software, which you see over this side on the right. So the hardware consists of a force transducer, a culture chamber that is sterile and can be autoclaved, while also being modular to allow easy access for cleaning and disinfection. But the slice is suspended between the force transducer and the actuator. So the actuator allows the tissue to shorten. The force sensor detects how strong the tissue it is, and the slice is suspended between them. If we look from a top view, it would look something along like that, as you saw in the previous picture. And you basically see that the slice is suspended between the force sensor and the actuator. We've also included a hole which is transparent for spectral-based measurements, such as, for example, laser or confocal microscopy, and so on. The advantage of the system compared to other commercially available systems is that it's sterile, it's benchtop, and it regulates its temperature, peristaltic pump, and flow perfusion on its own. So it does not need external equipment. It is pre-configured for fluid handling, as I mentioned. Measurements that are based on spectral measurements such as microscopy can be performed on it within the system without needing to take it out, and is much cheaper than any other commercial systems. It is also, because of its price primarily, it is it is sufficiently cheap to make it a high throughput system so that multiple parallel studies can be run in sync. With regards to the software, it allows data acquisition. It allows the simulation of different motion profiles on the myocardial slice, which can be physiological or pathological. And it also shows real-time data display, so real-time data of force, as well as sorting patterns and real-time data analysis. Finally, it can be fully automated, and endpoint assays, such as testing the response of the tissue to stretch, can be automated and require minimal user input. So these are some examples of the system in action. On this panel on the top right, you see the screen. You see a screen of the data display of the tissue. So what the myocardial slice is doing in this particular case, as before, it is producing force, which you can see on the side. You can see the tissue beating while also shortening. Now, the pattern of shortening in this particular case is this one over here. So this consists of two lines. So you will see that the slice is producing force, then shortening, then relaxing, and then filling again, as we said before, according to the force length loops. What the tissue is doing is shown in the blue line. What the model predicts is shown in the red line. By changing the parameters of the model, we can have the model predict a different shortening pattern that would correspond to physiology or pathology, respectively. And then with a click of a button, we can converge those two loops so that the slice, instead of doing the top shortening pattern, actually does a completely different shortening pattern. In this configuration and using the system, we're able to simulate a range of physiological and pathological work loops and study the effects of mechanical load on tissue remodeling in vitro. And this is just to show the point again. We have the same slice performing three different work loops. One in which is a physiological work loop with physiological parameters based on the model. One in which is a condition of high blood pressure, where you will notice that the slice is shortening much less. And one in which we sort of unloaded the slice and is shortening not only faster, but also to a greater extent, which you can also see on the right over here. Finally, one of the main advantages of the system is that not only can we simulate a range of conditions, but we can also track what's going on at the level of the tissue for prolonged periods of time. So in this panel, you see the force that the tissue is producing at any given time throughout a 72-hour culture. So in this particular case, we've simulated conditions of pressure overload, physiological load, volume overload, or an isometric load, which is the current status quo, and we've tracked the force production of the tissue throughout culture. One of the things you will notice is that after approximately 24 hours, the slices produce the same amount of force. So they plateau at around 24 hours, and they maintain a steady state. This can also be witnessed at the force length plane, as we said before, where initially we start with small loops, and then progressively the loops become bigger and bigger because of the way the model works, where more force corresponds to greater work loops and greater shortening.
The potential applications of myocardial slices are several, from cell integration for regenerative studies all the way to personalized medicine. The following part of this presentation will illustrate some of these applications. We utilize stem cell-derived cardiac myocytes that can be easily identified and functionally assessed. We obtained a GCAMP6F positive human iPSC line from Bruce Conklin's lab in San Francisco. We differentiate these cells into cardiac myocytes using standard methods, and we obtain regularly beating cells which are similar to other iPSC-derived cardiac myocytes, but spontaneously emit green light when stimulated at 470 nanometers. The video on the right shows the application of these cells in co-culture with HUVEC marked with a red fluorescent protein. We transplanted GCAMP positive cells on rabbit myocardial slices, and we performed optical mapping after 24 hours. The video shows that the GCAMP positive cells beat synchronously with the slice during point stimulation and field stimulation. Another important limitation of in vitro models is that they do not recapitulate injury, which is essential to recreate the conditions of application of regenerative strategies. we are developing approaches to simulate diseases in slices in vitro. One is cryo injury. This is simply obtained by touching the slice for a few seconds with a metal spatula, which has been kept in liquid nitrogen. Using MEAs, we observe complete conduction block across the injury. In addition, we observe a large number of alpha smooth methylectin positive cells after 3 days in culture. We developed another model of injury based on ischemia reperfusion. The simulated in vitro ischemia used a solution containing high potassium, no glucose, and a low pH. We perfused slices with the solution for either 30 minutes or 1 hour, and then we perfused again with oxygenated tyrode for 24 hours. The model is promising because it does not induce loss of cell viability in the slice, but a significant reduction in contractility even after 24-hour reperfusion, and this seems to be dependent on the duration of the ischemic period. Another project we have started is assessing myocardial slices for manipulation of gene expression. We obtained adeno-associated virus expressing EGFP, and we injected it into the tail vein of rats at 2 different concentrations. After 2 weeks, we prepared myocardial slices and imaged their fluorescence. At both concentrations, we observed a significant number of cardiac myocytes expressing EGFP with distribution across the whole thickness of the slice. We fixed myocardial slices cultured for 24 hours and then stained for DAPI, vimentin, EGFP-KEV3, and anti-GFP. We could clearly identify cells infected by the adenoviral vector expressing GFP in the context of the myocardium. We calculate that approximately 10% of all myocytes were infected. In summary, compared with other models, slices offer significant advantages. They present intact myocardial structure, they can be made from human biopsies, can be cultured under physiological and pathophysiological conditions, increase the throughput compared to whole heart studies, and allow investigation of mechanisms. Most importantly, they can be cultured for a longer period of times, opening the door to applications necessary for studying cardiovascular biology in a more relevant context. We would like to thank the cell electrophysiology team, in particular those who have contributed directly to this work. They are Eve Dries, Sam Watson, Filippo Perbolini, Brian Wang, Raquel Nunez-Toldra, Sam Kitanan, Iphigenia Bardi, and Oisin King. A special thanks to also Fotis Patoulis and Barrett Downing, who have also contributed to the delivery of this presentation.
Thank you very much to the audience for listening. I will be happy to answer any question.
Thank you very much, Professor Terracciano. This is Chris Matus again. Gentlemen, thank you very much for the excellent webinar. We now have time for questions and answers. So attendees, you can type in your question in the chat box, and we will get an answer to you. And for clarity, Dr. Terracciano's associates will repeat the answer. And so I have a question. As the questions come in, can you confirm that the data that you were showing with the MyoLoop was with human slice preparation? Is that correct? Or was that with rabbit?
So the examples in the movies with rat slices, but we have used human tissue easily, but we feel that working with human tissue when we get it is much easier. The problem for us is the scarcity of human tissue, so it's more difficult for us to have examples, but we can use it very easily. Does this answer your question?
Yes, thank you. Here's a question for you from the audience.
Please, can I say, please, you know I have a speech disability, So if anything is not clear, feel free to ask me to repeat, please.
Okay, so I understand. If it's not clear, I'll ask you to repeat. Um, how does the inclusion of pressure volume loop characteristics influence transcriptional changes in the cultured slices?
So this is work currently under investigation. As we speak, we have samples analyzed from transcriptional factors that are undergoing RNA-seq analysis. So we'll be able to answer your question very soon, hopefully.
So you're saying it's under investigation presently.
Yes. So we have
Okay.
all the samples and we are doing these experiments.
Okay, we look forward to that. The second question and comment from the audience, very elegant work. Have you characterized the model pharmacologically, for example, with sympathomimetics such as isoprenaline or phosphodiesterase inhibitors such as imovadan?
So we have extensively characterized the effects of isoprenaline on myocardial slices. We have used isoprenaline both as a positive inotrope and also as an inducer of arrhythmic behavior. Maybe you can repeat.
So we've used isoprenaline extensively in previous experiments, and we used it for which purposes did you?
For both inotropies and arrhythmic behavior.
So both for inotropic and arrhythmic behavior studies.
And the effects are similar to what we have predicted, which is positive inotropies and a higher level of arrhythmias. But what we observe, as expected for a loaded three-dimensional preparation, is a much lower KD for isoprenaline due to the higher sensitivity of this preparation, possibly reflecting much closer resemblance of in vivo conditions. Regarding the people bending, no, the answer is no, we have not had yet.
So for clarity, could you summarize the answer, please, Fatios?
So we've used the living up myocardial slice with isoprenaline extensively, but for the phosphodiesterase inhibitors, we haven't used that model just yet.
Yeah,
regarding isoprenaline, we find that it matches the in vivo conditions quite well. Better.
And regarding the myoloop experiments, we have not done that yet.
And you showed the diagram of the culture chamber, the desktop culture chamber. How long can you keep the slices viable in that chamber? You mentioned someone else did 4 months, but how long have you kept human slices in that chamber?
So our problem is that we only have a limited number of chambers, so time for too long it becomes a problem for us. So we only have time out to 3 days, but we don't know whether keeping it for longer may be possible. We suspect that because the preparation is fairly stable, we could probably keep it for many more days. Do you want me to repeat?
Yes, could for clarity, could your
So,
colleague repeat?
sure. Currently, we have used the human tissue for up to 3 days due to
Okay.
current constraints with the actual chamber setup at the moment. But we do envisage that we can keep it much longer.
Okay. There's another question from the audience. How do you know if the resting sarcomere lengths are changing over time in the slice preps? Do you measure it, and is it at physiological lengths?
So I think the question is whether the sarcomere length changes with time due to the physiological stimulation. So the answer is no, we don't know. We can measure it, but we cannot measure the sarcomere length. Say after 3 days, we have researched that this is certainly possible due to the modeling. And you
So I
may
understood that. Yeah. Sorry. I understood you said you don't measure it. Yeah.
yes, but I want to also add that we use a laser refraction method, which is very accurate and the most accepted way of measuring sarcomere length in tissue. But also, the people don't never measure sarcomere length. So we are already ahead of many other groups in the setting of this kind of measurement. So
I understand. Yeah. So it's possible. You're saying it is possible to do it.
Definitely.
It just hasn't been done. Is that
Yeah,
right?
exactly.
The next question is, did you assess the prevalence of cardiac fibroblasts in the tissue slice?
So we have published a paper in 2018 in cardiovascular research where we studied fibrosis in myocardial slices. We found that if the slice is cultured unloaded like in the standard method, there is a massive proliferation of fibroblasts, whereas if we keep the loading and almost sarcomere length, the fibroblast proliferation does not occur.
Okay, for clarity, could your colleague please repeat the answer?
Sure. So in unloaded conditions, there is an increase in fibroblasts in the slice model, and in loaded conditions, there is also so there's a decrease. In an overloaded condition, there is also an increase in the fibroblasts population. And in normal loaded conditions, there is no increase in fibroblast population within the living myocardial slice model.
And this is published in cardiovascular research.
In 2000, yeah. And this was published by our group in 2018.
No, by
this group, our group,
yeah.
sorry, excuse me. This was published by our group in 2018 in the Cardiovascular Research Journal.
Yeah.
Very good. Well, gentlemen, thank you very much for your participation in this webinar. It's very interesting and cutting edge research, and we wish you all the best. We look forward to more results coming from your laboratory. And thank you to everyone who attended and who will listen to this recording. And we wish you a good day, and stay tuned for the next webinar from AnaBios, which should be coming around in February. Thank you, everyone.
Thank you very much, Chris. Goodbye.
Thank you.
Thank you.
Bye.